Good afternoon, friends. Yesterday we saw taxonomy in relation to anatomy. Today we'll see embryology in relationship to taxonomy. What is the importance of embryology in taxonomy? Embryology is the study of micro and megasporogenesis, gametophytic development, fertilization development of endosperm, embryo and seed coats. Embryological evidences has been used for solving the taxonomical problems at almost all levels. These evidence have been resolved the doubtful systematic positions of several taxa. However, the role of embryology in solving taxonomical problems was first brought by German embryologist Scanarp in 1931. Uh, Another worker in embryology like Maheshwari, Bhojwani, Bhatnagar, Radford, they brought some basic embryological characters which have proved to be which have proved to be special basic embryological characters which are used in taxonomic considerations. They have been considered important, especially important in taxonomy. The first character is presence of type of another tapetum. Another is development of structure, position, and orientation of the oils. Uh, Megasporogenesis and development of embryo sac, type of embryo sac, forms of embryo sac, type of embryogeny, fertilization, and seed coat. Embryology, when you consider the first most character like of classification like dicots and monocots. Angiosperm are usually divided into dicots and monocots. The primary classification of angiosperm is based on the major embryological character, that is number of cotyledons. Another one is heliobate. These monocotyledons order treated as subclass in some recent systems of classification is characterized by the presence of helobial type of endosperm. That's why it is known as helobia. Orchidales. This distinguishable embryological character of the number of this order is the presence of undifferentiated embryo and very little or no endosperm. Podosteminaceae members of this family are recognized because of the formation of pseudo embryo sac, which is formed by nuclear cell. Another example is Lemnaceae. Phylogenetic studies indicate that Lemnaceae have been derived either from Helobels or Araceae. But on the basic embryological studies, Maheshwari suggested that Lamnaceae have been evolved from Ericaceae, not from Helobials. So he could separate it from the embryological studies. Crassulaceae embryological study suggested that it should be placed in order Rosals. Like these were some of the examples of embryology that played a significant role in solving the taxonomic problems. Embryology is the study of micro and megasporogenesis, gametophytic development, fertilization, the development of embryo sacs, and seed coat. Although the possibility of utilization of embryological characters in taxonomy was indicated by earlier botanists like Hofmeister, Strasbourg, the role of embryology in solving taxonomic problem was first brought earlier, I said, by the German botanist Scanarp in 1931. Embryological, what do you say, characters have been less prone to adaptive stress and relative stable and have acquired a great significance in plant taxonomy. They are considered a unique character to delimit taxa or to solve this taxonomic problem, especially when the external morphological characters are inconclusive and misleading as a result of convergence due to climatic or ecological factors. Thus, embryological evidence has been used in solving taxonomical problems that are doubtful for systematic positions of several taxas. However, some embryological characters have proved to be significant, helping in determine relationship with family, genera, and species, but less useful at the rank or order level, subclass or class. Maheshwari, on the basis of extensive work of embryology, he have provided that list of some family tribes, etc., where embryology has either supported earlier classification or has proposed a new systematic position for the taxon concerned. The embryological characters have been used in different ways, like in analysis of evolutionary trends, in circumscription of delimitation of taxa, in making decision on systematic positions, etc. Like some of the characters used, like basal placentation in RLAC, Nymphaceae, Oleaceae, Papiroaceae, Umbelliferae, Ranunculaceae, like bisolateral in Graminea, types of embryo. These are the types of embryo that have been seen, and like bent embryo in Anacardiaceae, Cactaceae, Leguminaceae, coiled in Potamogenaceae, Solanaceae, encircling in Malvaceae, Menispermaceae, Moraceae, some members of Amaranthaceae, Cactaceae, Nyctagenaceae, folded, linear, in various families, Araceae, linear in Amaryllidaceae, Olaceae, Rubiaceae, sagittate in Vitaceae, spatulate with endosperm in Asclepiadaceae, Birbidaceae, Oleaceae, Coronaceae, Osculadaceae. Uh, these are the types of embryo which were been used according to different families. The basic, these are the several embryological characters that has been proved for taxonomy significance earlier that we have discussed. Uh, another characters that have been especially important in taxonomy considered include the presence and type of anther tapetum, whether glandular, amyloid, number of arrangement of anther loculi, types of anther and endothecium, structure and thickening of endothecium, and the nature of tapetum, etc. Development of organization of pollen grains, number of position of germ spores, exam stratification. These are also some of the characters that were used. Megasporangium type of wall formation in megasporogenesis, arrangement of megasporogenesis, position of the megasporogenesis, development of embryo sac that we have already discussed. Application of embryological data in taxonomy, the delimitation of angiosperm is the first instance that has been largely based on embryological characters. These characters have been, they are characterized by enclosed ovule that are mostly anatropous, polygonum types of embryo sac is in the majority of flowering plants, double fertilization, triple fusion, and post-fertilization development of polyploid endosperm, which also support the probable monophyletic origin of angiosperm. The further primary classification of subdivision of angiosperm in two classes, namely monocot, dicot, that are based on embryos and almost constant for the group. However, certain groups are well marked out by their embryological features that can easily be identified. These members, the exception in particular are taxonomic significance. Uh, there are a few orders in angiosperm, the members which are morphologically well defined and also exhibit a set of uniform biological features. For example, some of the families of Carophylls, it is widely known as Centospermi and is embryologically very distinct. This order is characterized by trinucleate pollen, by endemic crasonucleate ovals, which are campylotropous or amphiotropous, commonly with the inner integument longer than the outer one, very often with the space between integuments to the chelazal end. The seeds of these are curved peripheral embryo, more or less surrounded by food storage tissues, which consist mainly of perisperm with little or no endosperm. Uh, Another Ericales, this is a homologous group of Sympatelis family, including member, most of which share several common embryological features, including the production of pollen in tetrads. Another order is Ginchanails. It is characterized by simple, most opposite leaves. Integument is there, lack of integument, tapetum, and nuclear endosperm, which helps it to be distinguished from the other Sympedalous order. Already we have discussed the Halo B. This order under the monocotyledony, according to Engler, it is characterized by halobial type of endosperm. Although recent systems have raised the order to the level of subclass, the relationship of the constituent order is scarcely disputed. Orchidales, we already discussed undifferentiated embryo with little or no endosperm. Some of the characters that are below the family level embryological evidences that strongly support the family in genera. The few examples are Onagraceae. This family is characterized by the presence of peculiar monosporic four nucleated onagrade type of embryo sac, and it is all in its genera except Trappa. Such of the embryo sac is derived from micro pilar microsporangia or the tetrad. The lower three chelazar microspore do not disintegrate. In this type of embryo sac, antipodals are absent. Different systematics have placed the genus Trappa in several other ways. Some have placed it under Onagraceae, some have placed it in the isolated member of Holagraceae, another have placed in genus monophyletic family Trapezae, so on. The embryological evidences, however, strongly support the last view. Trappa has a well-developed spring-like that is known as suspension rostorium. It, in addition to the 8 nucleated embryo sac, which are absent in any other member of Onagraceae. Further, in the Trappa, the ovary is semi-inferior uh, bi bilocular with one oil in each locule, and the fruit is large, one-shaped, drupe is present. Another example was Empitraceae. These families closely related to Euphorbiaceae and Celastraceae. Uh, the family of Monoclamidae. Later, several taxonomies thought that they are related to Ericaceae on the basis of several morphological features. The later view has been supported by this Samuelson on the basis of embryological data. Cypressae, earlier systematic considered the family Cypressae and Poaceae to be closely associated. However, morphological and anatomical features and the structure of spikelets revealed that the two families are quite distinct. These two families alliances is also contradicted by embryological findings due to the due to some of the findings that like microsporangia is of simultaneous type in Cypressae and tetrad members do not separate, whereas in Poaceae, microsporangia is a successive type and tetrad members separate. The partial cell is present in Archosporium of Cypressae, while it is absent in Poaceae. Antipodals are ephemeral while they are complex and becoming uh, conocytic in Poaceae. These are some of the examples that distinguish the Cypressae and Poaceae. Other families also, the Liliaceae, there has been a rearrangement of tribe and some families within the Liliaceae based on embryological studies. A large number of genera also have been assigned their position on morphological basis, embryological ground. Liliaceae and Embryology are considered as polyphyletic families, and certain groups of these families are related to each other based on resemblance. Resemblance. Hutchinson has united three types of subfamily Draconiidae and UKC. There are other examples also of Poinia genus that are monogeneric type Poniaceae family Ranunculaceae. So we have seen some examples of uh, families, characters that were significant for taxonomy. What is the importance of embryology in relation to taxonomy? Why is the embryology important for study? in taxonomy. In the next, we will continue the next point. The next lecture, we will continue the next point. We will stop here.